Hello, and welcome to the chapter when we discuss how the DNA genetic code is used to make proteins for your body. Some goals and objectives we're going to be covering during this lecture are: number 1, explaining what the central dogma of protein synthesis is. Number 2, we're going to be describing the genetic code itself. 3, we're going to list the steps in transcription. We're going to also describe the different steps in RNA processing. And last but not least, we're going to list the steps in translation. Okay, so there's transcription, RNA processing, and translation. The last thing is we're going to basically connect all of these different steps so we can understand how proteins are actually made from DNA itself. So let's do a little recap on the organelle of the nucleus. Remember, the nucleus is the actual cell's control center, and that is where the genetic code, aka the DNA, is actually maintained within the nucleus. When the DNA is very, very loose, when it's very loose coiled DNA strand, that's actually known as chromatin. It's not known to be a chromosome until that loosely wound DNA is very, very tightly wound around the histone proteins. And then that's when we get the characteristic X-looking chromosome right here. So once again, before it gets nice and tightly compact, it's known as chromatin. After it gets really, really tightly, nice and compact, then it's known as a chromosome. So as you can see right here in this picture, you actually do see here's a loose DNA wrapped around these histone proteins. That's why you see the labeling of chromatin right here. Okay, the very center where these two chromosomes are actually connected, that's called the chromosome kinetochore. Okay, this is also the area where those spindle fibers are going to come and grab on to each of the sister chromatids when they pull them apart, okay, during mitosis or meiosis. So the DNA strand consists of a chain of 4 nucleotides. Those nucleotides can be known as the adenine nucleotide, thymine, the guanine nucleotide, or the cytosine nucleotide. These are also the nitrogen base pairs here. Okay, so adenine, thymine, guanine, and cytosine are all the nitrogenous bases for DNA. When we're talking about how DNA is organized, we're going to talk about them in distinct sets of nucleotides on a chromosome, and these sets of nucleotides are known as genes. So basically just imagine you have a very, very extremely long strand of a bunch of DNA nucleotides, okay? Every single segment, every particular segment, and it could be, let's say, 1,000 base pairs, or it could be only 50 base pairs. It just depends on that particular gene and how long or how many amino acid base pairs there are included in that single segment of DNA, aka known as a gene. The genes of DNA are what actually control the characteristics that offspring will have by transmitting the info and sequences from the parent to offspring. Because remember, you're actually getting particular sets of genes transferred or basically inherited from your mom or from your father, one or the other. And let's do a little recap as well. Remember, so this right here, these light blue colors, that's a gene. That could be, let's say, the gene for hair color. But there's 2 different alleles to that gene. So let's say this is the brown hair color, which is the homozygous dominant letter. But if it's a homozygous recessive allele right here, maybe that might be the blonde hair color allele. Same thing right here. This is going to be a different gene. Let's say this is the gene for eye color. Maybe this is the gene for height. They're just basically a segment of DNA in the entire genetic code of an individual. Nucleotides themselves are actually arranged in codons. A codon are 3 base pairs put together. For example, when we have thymine, adenine, and cytosine in an actual codon, that's going to code for the amino acid tyrosine. Just like we have guanine, and thymine right here, this particular sequence of these amino acids for this codon, this actually codes for glycine, which is an amino acid. What are amino acids exactly? So you know how we talked about how we have a nucleotide, a nucleotide, a nucleotide. Nucleotides are actually monomers of the big polymer nucleic acid. Okay. Well, here we have amino acid, amino acid, amino acid. A bunch of amino acids are in a chain, a big long chain together, and those are actually what creates a protein. So amino acids are monomers of the polymer protein. Also, something to note here is that amino acids are actually held together by peptide bonds. That's very important to know because of the fact that peptide bonds are only found in this amino acid chain, in the chain of proteins. So these are all the 20 different amino acids that there are found today. Lysine is right here. Arginine is right here. Because remember, every codon codes for a different amino acid. The only thing, if you notice, they all have their root the exact same. They all remain the same right here in this root section. The only thing that's different is this R function right here. This little blue area, that's the only thing that changes in all these amino acids throughout. So this is all a list of all the different amino acids available for us and the different code sequences or codons for each. So the way you read this is, let's say I'm giving you that I'm telling you that the actual codon is AUG. So the first nucleotide in the order I gave you was A. So here's the first one. You're going to go to the A's. Then you look up here. The U is right here. So I'm going to go A, U, and I know it's going to be in this section. And then I just look for the G right here. So A, U, and here's our third nucleotide sequence. Here's my G, AUG. So this particular codon is actually coding for methionine. This is also the start codon. See this right here? There's one start codon and there's 3 stop codons. What that means is when your body is actually able to read the code AUG or methionine, it's actually telling your body to begin making that protein starting right there. It also gets a code to stop protein synthesis at a particular portion of the DNA strand or gene. And that's what these guys do right here. These 3 codons do right here. UAA is a stop codon, UAG is a stop codon, and UGA. So whenever you get to these 3 codons, you know that is a particular spot of where you're going to actually stop creating that protein you're working on. So the genetic code itself is the chemical language of DNA instructions. And this is what we're going to be looking at today. Sorry about that. This is what we're going to be looking at today. We're going to be looking at how we go from a DNA strand or template, how we undergo transcription to create this messenger RNA strand right here. And it's messenger RNA. Remember, there's a difference between nitrogenous bases of DNA and RNA. Remember, RNA has uracil, which we can see right here indicated by these U's. Okay, so we actually transcribe DNA strand into messenger RNA. Then from a messenger RNA, we translate this code, this actual codon, into a protein, basically an amino acid. These amino acid, amino acid, amino acid. And these amino acids are connected by a peptide bond right here. An entire sequence of amino acids are what's creating a protein, which is our final product. One thing good to know is the central dogma of molecular biology. This is defined as each gene in DNA carrying information needed to construct only one protein each. So one gene equals one protein to synthesize. So once again, just to recap, we're actually going to see now the actual steps it's going to take for us to go from the DNA, which is, at this point you should know DNA is found in the nucleus. So we're going to get DNA from inside the nucleus. We're going to transcribe it, undergo transcription, so we're transcribing the DNA, because DNA, one key here, DNA will never, ever, 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 ever leave the nucleus. So that's why we have to transcribe it. We basically have to copy the code into messenger RNA, and then it can actually we can actually retract that information from the DNA itself through messenger RNA itself. So it's not DNA leaving the nucleus. Once again, DNA will never, ever, ever leave your nucleus. Because think about how many mutations or how much things can occur if DNA left your nucleus. That's why it's protected by that double membrane. It's not only protected by the plasma membrane around the cell, then also it has a double membrane around the nucleus protecting your DNA, because if anything goes wrong with your DNA and a cell gets replicated, that can ultimately lead to cancer. So we go from DNA inside the nucleus, we transcribe that information and create a messenger RNA strand. Then that messenger RNA strand actually gets translated, because now it's basically we need to translate the information into something that your body can read. So we translate that messenger RNA information into proteins. So let's go ahead and check out how we do that. And before we go ahead and continue on, I did want to recap once again on DNA versus RNA so we understand the difference between coding of these two individuals. So DNA is a double helix form. RNA is mostly single-stranded. DNA stands for deoxyribonucleic acids or nucleotides. Then there's also RNA, which is ribonucleic acids or ribonucleotides. DNA is actually made up of the sugar deoxyribose, whereas RNA is made up of ribose sugar itself. The nitrogenous bases for DNA is adenine, guanine, cytosine, and thymine. RNA nitrogenous bases include adenine, guanine, cytosine, and here's the kicker, here's the change here. Instead of thymine for RNA, we actually have uracil. So if you recall, in DNA over here, do you remember what actually pairs with adenine? Adenine will always pair up with thymine, right? As you see here. So if we have RNA and we switched the thymine out for uracil, now we have adenine pairing up with the uracil. So that's a big difference here as far as the pairing of the nitrogenous bases. Okay, the function of DNA is to store genetic information inside of your nucleus in your cell. The function for RNA is to actually make proteins, and then it also has a couple other roles as well. So the very first step we're going to be looking at today in transforming DNA into proteins is going to be transcription. One thing you want to remember is that transcription occurs inside of the nucleus. And there's 3 steps to transcription. There's an initiation step, an elongation step, and a termination step. So the first step, once again, occurs in the nucleus. The first step is initiation. The first step is basically going to be when RNA polymerase attaches to a promoter sequence. So RNA has to come along and it'll actually attach to DNA itself, but it has to look for a specific sequence so it knows where to start. So I think of the ta-ta box like somebody holding a flag out in the air saying, hey, start here. You're going to have to start right here. That's what the ta-ta box is. So your body actually has that code set up specifically for RNA polymerase to come and attach to it. So it knows exactly where to start initiating transcription for your body. So after it's actually began, or the RNA polymerase is actually attached to that DNA strand, then it's going to basically unzip the DNA strand open, because it has to be able to copy that DNA code and transcribe it into RNA. So now, before it can actually, after it unzips it, now it's actually going to be looking for that start codon. Remember what the start codon is? It's AUG, which actually codes for the amino acid methionine. So now it's going to be looking for the AUG. Once it finds that AUG code, it's going to begin reading the DNA code, and now it's going to start adding the RNA nucleotides that are complementary to that particular DNA strand. So right here, we're looking at, once again, this big, huge purple blob is RNA polymerase. Remember, it came and it attached to the promoter sequence, or the ta-ta sequence of the DNA strand. That also allowed it to unzip that strand of DNA. Then after it unzipped it, now it's allowing it to actually start laying down RNA nucleotides that are complementary to that DNA strand itself. Remember, we're making RNA here that's complementary to DNA. So here we're going to have uracil instead of thymine on our new messenger RNA we're creating. So remember, it's elongating this way. So in other words, first it started off with 1, 2, 3, 4, and as it started laying down more and more and more RNA nucleotides, this thing's just getting longer and longer and longer and longer until it's going to reach the stop codon, or one of the 3 stop codons, and then that's when actually transcription will actually cease. And they'll actually stop, and now this messenger RNA needs to get processed before it can go through the next step. So this is actually looking at elongation occurring. So remember, this is where I said all the RNA strands are getting laid down. Okay. And this is the termination right here. Remember, this is where it started in that ta-ta box sequence. Then it actually found the start codon, the AUG. It began transcribing, transcribing going this way right here. And then at the very end, after that messenger RNA is being grown, and this right here, this red part, is actually indicating to us the stop codon, basically the stop site, telling us that there's a terminator region there, telling us, okay, we're done coding, we can stop now. So this is stop, once again, our promoter sequence, that ta-ta box, and then we have a terminator sequence right here somewhere. So, and this length of the gene itself will actually be different for every gene. So one gene might, eye color might be 100,000 base pairs, and let's say hair color might be something completely different. So, at the very end, you should be left with a pre-immature messenger RNA. It's an immature RNA because it hasn't been processed yet. Before this last little tidbit on the 3' side of the new pre-messenger RNA, before it can be released completely from this DNA strand from where it was coding, there's a sigma factor that has to come and rebind to RNA polymerase, and that causes it to bloop, and kind of pop off and allows this messenger RNA to go on and continue the rest of the process that we need it to. So this is what we just went through right here. We went through transcription, okay? So transcription converts information in a gene to RNA. So basically we went from DNA inside of the nucleus, a particular segment of DNA, which is a gene. So we went from that, we transcribed the information using RNA polymerase, and we ended up with a messenger RNA, or you can say an immature RNA because it still needs to get processed, okay? This is a really good link of a video you should watch at this point and make note of, so that way you can kind of follow along and make sense of every single one of these steps. The next part we're going to get into is actually in between both of these parts. Remember, there's transcription and translation, but we actually have to change this messenger RNA before it can continue on and we can continue translation, okay? So let's go ahead and take a look at what needs to get done to that premature messenger RNA first. Right here is premature messenger RNA. It has to be modified before it can actually leave the nucleus. So right now we're still in the nucleus, remember, because the DNA got transcribed into pre-messenger RNA, and that's why we're still in the nucleus right now. But it cannot leave the nucleus until this is done. So what happens here, and we're just pretending like this messenger RNA right here is actually just laid out right here completely long. So just imagine this long green strand is right here all long and green. What happens is there's actually exon portions and intron portions, okay? The exon portions are the portions that have expressed gene information. That is why we're going to keep those expressed segments, and we're going to get rid of our intron segments. Our intron segments are non-coding sequences, so we don't need them. So they're disposable. So we actually splice them out using spliceosomes. Literally, we're splicing away. Get rid of that intron, get rid of that intron. This is called RNA processing when we're doing this. And we end up with just exons, which are the expressed segments of the gene. So now you can see exon 1 is now connected to exon 2 and exon 3 right here. And the final step of RNA processing is we actually have to add a 5' cap on the 5' side of that messenger RNA sequence. And then on this side right here, we actually have to add a polyadenylated tail. So we just call it a 3' poly A tail. The reason it's called a poly A tail is only because it has tons of adenine at the very, very tip of it. The reason it needs that adenine, or a bunch of A amino acids at the very end of it is because we want to make sure that if anything gets cut off by enzymatic activity when it leaves the nucleus, that it's not getting rid of any kind of the expressed gene information we need. So we'd rather it cut off this end and that end if an enzyme came across it. So it's kind of like a protection barrier. So once again, messenger RNA contains these introns, which are non-coding segments. So those are the ones we're going to splice out. with spliceosomes, and the exons are the expressed gene information. That's why we're going to keep them, and it's going to stay in the messenger RNA. The 5' cap goes on the 5' side. 3' poly A tail is a long strand of adenines. And now you just went through RNA processing. So now you are a mature messenger RNA. So now you are now allowed to leave the nucleus and enter into the cytoplasm of the cell. So we're coming here, we're leaving the nucleus, it got processed, and now this right here, this bubble was all the nucleus. So now we're allowed to leave the nucleus, and now we are a mature messenger RNA in the cytoplasm of the cell. Now we can go ahead and go on to the next step. First, we went through transcription, we went through RNA processing, now we're going to begin translation. Translation also has 3 different steps. It has an initiation step, an elongation, and a termination step. You must have a mature messenger RNA for this portion. Otherwise, there will be issues, and more than likely, the protein synthesis will actually cease, and actually just, you'll have a lysosome come and engulf it because it's actually been defaulted at that point. But if everything went good, we did our RNA processing correctly, and now we're in the cytoplasm. Now we can go ahead and begin looking at translation. First things first, let's go ahead and look at some things that we're going to need to know about with translation. Do you remember what ribosomes are for? Ribosomes is the organelle inside the cell, and it's what gives rough endoplasmic reticulum its rough look. Remember, ribosomes are meant for protein synthesis. So here's where ribosomes are coming back into play. When you see a little t in front of the RNA, that's standing for transfer RNA. It's actually going to be carrying amino acids right here at the very tip of it when we're undergoing translation, and we'll see that in just a second, or you would have seen it in that video in the previous slides. Amino acids, remember amino acids like cytosine or glycine, they're actually, or tyrosine, they're basically coding, when you put amino acids together with peptide bonds, it codes for an individual protein itself. So here we go, beginning with translation. There's going to be an initiation stage first. That's when messenger RNA actually binds to a small ribosomal subunit upstream of the start codon. So here's our start codon, or AUG. Here's our small ribosomal subunit right here. Okay. Then after that happens, we actually have an initiator transfer RNA anticodon, which comes along and attaches its anticodon, because remember, we have the codon, remember the AUG, over here connected onto our messenger RNA. But we need an anticodon, which is going to code and attach and complement our codon. So that's why we have a codon here. We need an anticodon to connect. Remember, opposites attract, okay? So once again, first we start off with our small ribosomal subunit that comes in. It attaches upstream of the start codon to our messenger RNA. Then after the small ribosome attaches, now we have our transfer RNA come and bring its anticodon right here. And that is going to go ahead and attach right here. And if you look right here at this purple hexagon, that is actually pretending to be an amino acid. Because remember, transfer RNAs carry amino acids individually. Okay, so that's what we're looking at right here. Then, so once again, our codon gets attached to an anticodon, which is carried by transfer RNA. After that happens, guess what comes into play? Our last piece of our ribosomal subunit, because a ribosome actually has 2 parts. It has a small portion or subunit and then a large subunit. So now we have the large ribosomal subunit come and join the small one, and this makes all together the translation complex right here. One thing I want to point out to you are these 3 different sites you see right here on this ribosome. There's an A site, a P, and an E site. If you notice, our initiator transfer RNA right here, it actually starts in the P site. This is the only one out of all of the transfer RNAs that's going to start here. Okay, and what I like to think about when I think about the EPA sites are A is for arrival, P is for polypeptide growing, E is for the exit. And I'll show you what I mean in just a second right here. So here we are. Here's a small ribosomal subunit again. Here's our messenger RNA. Here's our codon right here for the start codon. Here is our anti-codon, which is actually carried on our transfer RNA, which is also carrying an amino acid right here. So what happens is, here's our initiator one. It comes in, it attaches to the P site of the large ribosomal part. Then our next or second transfer RNA comes in, and it's going to attach to the arrival site. And they kind of shift over and move down like a conveyor belt. So that A comes in, it's going to scoot both of them over. This one right here, the initiator transfer RNA, is going to go over here to the exit site. And then it's going to end up exiting out after it actually dropped off its amino acid here. So an amino acid here, like I said, the next one comes in, it's going to attach its amino acid to the one that was before it, and then it exits out through the exit portion on the E side over here. So it's going to come in, attach its next amino acid, growing this polypeptide chain, and then it exits out. Then the next one comes in, drops off its polypeptide to grow this chain, and then it exits out. Okay, the polypeptide is going to grow and grow and grow until the very end. Okay, and we'll look at that next step. This is going to be the termination step right here. An amino acid chain, aka what's known as a polypeptide, is finally released when a stop codon is reached. So there's going to be, let's say for example right here, a stop codon. You can read it right here. It's coding for stop. So then what happens is a release factor right here comes and attaches into the arrival site. It terminates translation completely, and the components can now just break apart and break off, because we really don't need these ribosomes right here anymore. We also don't need the transfer RNAs anymore, and this is what we really care about. This is our end product. This is our polypeptide, or our aka our protein. So remember, here's an amino acid, here's a peptide bond, an amino acid, peptide bond, amino acid. All this together makes a big long chain, which codes for a protein that's needed in your body. So this is once again the codon, just to refresh your memory again, that codes for the amino acids. So we have UUU, which codes for this abbreviation of phenylalanine. We have UCU, which codes or is a codon for serine. We have UAU right here, which codes for tyrosine. Also UAC codes for tyrosine. Here's our 3 stop codons, which you would want to know. And here, last but not least, is our methionine codon, which is AUG. So to recap from beginning to end, remember we actually have the DNA that's in our nucleus. So this first part that has to happen that occurs inside the nucleus is known as transcription. Transcription right here begins with RNA polymerase recognizing a promoter sequence, which is known as the TATA box. Once it recognizes that TATA box, it's going to actually begin dropping down RNA nucleotides in a 5-to-3 direction to create a premature messenger RNA until it reaches a termination sequence. Once it reaches a termination sequence, it'll actually have a release factor that comes in and attaches to RNA polymerase, and that causes everything to disassociate and releases our immature messenger RNA for us. Once we have that messenger RNA right here still inside the nucleus, we need to process it next. So now we're going to undergo RNA processing. Remember, RNA processing, what happens is we have to actually splice out the introns or the non-coding sequences on this messenger RNA. The coding sequences that we keep are the exons, because they're the expressed parts of the code. And then the final part of processing the RNA is going to be to add a 5' cap and a 3' polyadenylated tail here. Now we can actually consider it a mature messenger RNA, and it can actually leave the nucleus at this point. After we have undergone transcription in the nucleus and then RNA processing in the nucleus to get it out of the nucleus, now we can actually translate that information over here in the cytoplasm of the cell. First things first is we have a small ribosomal subunit that's going to attach to the messenger RNA. Then we have an initiator transfer RNA, which comes and attached to the codon, the start codon sequence AUG, and it carries along with it an amino acid. The reason it knows where exactly to attach to the AUG is because it has an anticodon, which is going to attach complementary to that messenger RNA. Once it attaches in the P site, because remember, only the initiator transfer RNA will attach to the P site, then we actually have all the rest of the transfer RNAs come in and attach in the A sites, or the AKA arrival sites. Then remember, it acts like a conveyor belt. It's going to scoot over, and then it gets to the polypeptide chain growing site, where it's going to drop off its amino acids to attach to the next one. And then it finally pushes over again. When another one comes into the A site, it gets pushed to the exit site and leaves out. The very end, it'll actually keep continuing translation until it reaches a stop codon, or one of the 3 stop codons we looked at before. Once it reaches that stop codon, it's going to actually disassociate completely with the release factor attached in the A site. The last thing to note is that even though we already made that protein, it can actually be modified after that into the Golgi, into its final form for any use that your body needed that particular protein for. So everything we just talked about how we go from DNA all the way to protein, it all occurred, remember, in the nucleus, and then also right here in the rough endoplasmic reticulum, where translation actually occurred. Because remember, these are where the ribosomes are found, right here, and that's why the rough endoplasmic reticulum actually looks rough, because of all the little dots, or all the little ribosomes that are attached to it. Then, like I said, the protein can be modified in the Golgi apparatus, which is the next organelle here, and it gets processed, remember, because the Golgi is kind of like a post office. It's going to receive a package, it's going to repackage it and find out where it needs to go, and then it's going to send it off on its proper destination. So that's what the Golgi is doing. It's receiving the actual proteins, and then it processes it, modifies it, and then sends it off to wherever it needs to go. So it can get sent out in a secretory vesicle outside of the cell. It can actually get transferred out over here. Maybe it needs to actually get killed because it wasn't processed correctly. So that's when a lysosome comes in. Remember, the garbage men, they come in and they eat any kind of cell debris, so it could possibly have that route. Lots of different routes that this Golgi can have that protein take. But the ultimate goal is to transfer it out to its proper destination.